Today, we'll be discussing on the history and scope of microbiology. So before that, we'll define microbiology. Microbiology is a science that deals with the study of organisms that are too small to be seen by the naked eye. Under this, microorganisms include viruses, bacteria, algae, fungi, and the protozoa. As we all know that microbial life has since shaped our atmosphere, our geology, and the energy cycle of all ecosystem. But it was only after the development of microscope and the various observations made by various scientists that led to the discovery of microorganisms. Microscopes are optical instruments that serve to magnify objects so small that cannot be seen clearly by unaided eye. So next, we'll discuss about the discovery of microorganism. Anthony von Leeuwenhoek was the first person to observe and describe microorganisms using a microscope of his own design and made one of the most important contributions to microbiology. Leeuwenhoek's microscope consists of spherical lenses mounted between two small metal plates. And also, he illuminated his liquid specimen by placing between two pieces of glass and shining light on them at 45-degree angle to the specimen plane. This provided a form of dark field illumination. Leeuwenhoek observation was reported to the Royal Society of London in 1673 with accurate shape and detailed movement. The organisms that he observed were possibly bacteria and the protozoa, which he termed it as animalcules because these bacteria and protozoa they behave like the animals. So next, we'll come to the conflict over spontaneous generation. The belief in the spontaneous formation of living beings from non-living matter is known as the doctrine of spontaneous generation, or it is also known as the theory of abiogenesis. Spontaneous generation was a widely held belief throughout the Middle Ages and into the later half of the 19th century. But this has no scientific basis. This view was finally challenged by an Italian physician, Francesco Redi, who carried out a series of experiments on decaying meat and its ability to produce maggots spontaneously. Redi placed meat in 3 containers in his experiment. One was uncovered, the second one was covered with peppers, and the third one was covered with fine gauze that would exclude flies. Maggots developed in the uncovered meat from the eggs laid by the flies. The other 2 pieces of meat did not produce maggots spontaneously. However, flies were attracted to the gauze-covered container and laid their eggs on the gauze and produced maggots. From this, the development of maggots resulted from the presence of flies' eggs and not from the meat spontaneously. But the debate and the concept of the spontaneous generation was revived in 1748 by an English priest, John Needham. John Needham, in his experiment, he boiled mutton broth and then tightly stopped the flax. Many of the flax became turbid because of the presence of microorganisms in it. He thought that organic matter contained a vital force that could confer life on non-living matter. Another scientist came again later, Lazzaro Spallanzani, an Italian priest and naturalist, improved on Needham experiment. He sealed the flask that contained water and seeds and placing in boiling water for 3/4 of an hour. Here, no growth took place as long as the flask remained sealed. He proposed that not only air carried germs to the culture medium, air is also required for the growth of animals that are already in the medium. Further, the controversy over spontaneous generation was resolved by a French scientist, Louis Pasteur, in 1861. In his experiment, Pasteur first filtered air through the cotton and found that objects resembling plant spores that had been trapped. If this piece of cotton is placed in a sterile medium, microbial growth will appear. Pasteur undertook another experiment. In his experiment, Pasteur boiled nutrient solution in a flask which have a variety of curves while keeping the ends of the necks open to the atmosphere. In this experiment also, no growth took place when the flask is cool, even though the content of the flask were exposed to the air. He pointed out that no growth appeared in the flask because the germs and the dust particles are trapped on the walls of the curved necks. If these necks were broken, growth will commence immediately. Finally, a final blow to the spontaneous generation was given by an English physicist, John Tyndall, in 1877. He demonstrated that dust indeed carried germs, and if these dust were absent, growth remained sterile, even if directly exposed to air. He also provided an evidence for the existence of heat-resistant forms of bacteria. Tyndall then developed a method of sterilization by discontinuous heating, which is known as Tyndallization. This process of Tyndallization could kill not only the growing bacteria but also their heat-resistant forms. Now, we'll discuss on the role of microbes in diseases. Earlier, many scientists suggested that diseases were caused by invisible living creatures. But this idea was not accepted and people believed that diseases was due to supernatural forces. On this, Louis Pasteur postulated a theory that is known as a germ theory of diseases. Germ theory of diseases states that infectious diseases are caused by microorganisms. But he failed to prove the theory. To prove this theory, that is the germ theory of disease, the first direct demonstration with evidence of the role of bacteria in diseases was provided by Robert Koch from the study of anthrax. Anthrax is nothing but it's a disease caused by a particular type of a bacteria. Koch established the etiological role of bacteria in anthrax and proposed a set of rules in the establishment of etiology. The procedures made by Koch came to be known as Koch postulates. So we'll see the Koch postulates, what it states. Koch postulates states that the microorganisms must be present in every case of the disease, but it should be absent from the healthy organism. In the second point, he states the suspected microorganism must be isolated and grown in the pure culture. In the third postulate, he states the same disease must result when the isolated microorganism is inoculated into a healthy host. Finally, in the Koch postulate, the same microorganism must be isolated again from the disease host. Koch not only postulates this germ theory of disease but also developed media that is suitable for the growth of bacteria isolated from the body. And this has resulted in the development of neutron broth and neutron agar, which are in use today for the cultivation and growing of different forms of bacteria. Not only Koch study on the microbial role of diseases, but Pasteur's also contribute in the field of microbiology. His contribution in this field of microbiology is numerous. For the prevention of diseases, vaccines against rabies and anthrax were developed by Pasteur. During his studies on chicken cholera, he discovered that culture lost their ability to cause a disease when incubated for long intervals. If these chicken were injected with this attenuated culture, here when we say attenuated, it means the weakened culture. They remain healthy but develop the ability to resist the disease. So such an attenuated culture is termed as vaccines. Another scientist named Joseph Lister developed a system of surgery which is designed to prevent microorganisms from contaminating wounds. Lister implemented the use of sterile surgical instrument and used carbolic acid, which we know as phenol nowadays, during surgery and wound dressing. Now we'll see the microbial effects on the organic and inorganic matter. Here, when we talk about the microbial effects on organic and inorganic matter, there is one term that is known as fermentation. We'll first define this fermentation. Fermentation is nothing but it's an energy yielding process in which organic molecules serves as both the electron donors as well as the acceptors. So earlier, fermentation was believed that it was due to the chemical instability that degrades sugar to alcohol. Although Theodor Swan and others proposed in 1837 that alcoholic fermentation was carried out by yeast cells. But it was Pasteur who demonstrated that fermentations were due to activities of specific yeast and bacteria. Pasteur's discovery came from the work of wine souring, which resulted in the decline of alcohol yield. He showed that fermentation was failing because the yeast that is normally responsible for alcohol formation had been replaced by microorganisms producing lactic acid rather than ethanol. Pasteur's discovery on fermentation not only led to the study of wine diseases, but also he developed a technique that is known as pasteurization for the storage of wine. As we all know that microbes were the first living organism on our planet and are more numerous than any other kind of organism. From among the five kingdom classification proposed by R.H. Whitaker in 1969, microbes include three of the kingdoms, that is, Monera, Protista, and fungi. The subject microbiology deals with five major groups of microorganisms that are viruses, the bacteria, the fungi, the algae, and the protozoa. This subject microbiology has a great impact on medicine, agriculture, food science, ecology, genetics, biochemistry, and many other fields. So the subject microbiology has two main branches that includes the basic microbiology and the applied microbiology. So now we'll see what this basic microbiology deals about. The basic branch of microbiology is the study of biology of microorganisms, and that includes bacteriology, which is concerned with the study of bacteria. Mycology is concerned with the study of fungi. Algology is the study of algae. Protozoology is the study of protozoa. Microbial cytology is concerned with the study of microbial cell structure. And microbial physiology is concerned with study of microbial metabolism and growth. Next, microbial genetics is the study of genetic information of microorganisms and their regulation and function. Further, microbial ecology is concerned with the study of microorganisms in their natural environment. Microbial taxonomy is concerned with study of microbial classification. And finally, this biochemistry is concerned with the study of microbial enzymes and their chemical reaction. The applied aspect of microbiology deals with practical applications of microorganisms to solve problems that are related to diseases, water and wastewater treatment, food spoilage, and food production. So, that was all about the history and scope of microbiology. From the above discussion, we come to know that the science of microbiology involves microorganisms that are very small and cannot be seen by our naked eye. The subject microbiology developed only after the invention of microscope. And for this microscope invention, Antony von Leeuwenhoek showed microorganisms, which he referred to as animalcules, to the world for the first time. Simple organisms are developed by spontaneous generation was universally accepted prior to the introduction of microscope. So this theory, the spontaneous generation, was challenged and disproved by experiments of Francisco Redi, Louis Pasteur, and many others. The idea of spontaneous generation was displaced by another theory that is known as the germ theory of diseases. So, the tremendous strides made by infectious diseases research since the days of Robert Koch have led to the great expectation of infectious diseases control community.